Hi, I'm Ryan.
I'm Cade.
I'm Emily, and this is Lab 12, Color, Vision, and Perception.
The human eye system is amazing, and it's how we see and process and interact with the world. And this lab is to examine a lot of both what we see with our eyes and then how our brain actually processes a lot of the signals that we get from the things that we can see, and to test the limits of both what we can see and what our brain perceives from our vision.
Humans have what is known as binocular vision, meaning that our vision is formed from two separate imaging systems, one for each eye. If our brain didn't select one image to prioritize, our vision would look like this image of this car, in which one picture is spatially separated or by a certain amount of stereoparallax from the other, and all of our pictures would be overlapped. Instead, our brain selects one image to prioritize. In order to figure out what your dominant eye is, you can hold up a piece of paper with a hole in it and focus on a distant target with both eyes open. Close one eye, close the other eye, and whichever eye you can still see the target through is your dominant eye. Both myself and Emily are right eye dominant, while Cade was left eye dominant. At the back of the human eye, there's the optic nerve, which transmits the sensory information from the photoreceptors to the brain. The optic nerve doesn't have any photoreceptors on it itself, and as a result forms a blind spot. This blind spot is located about 15 degrees towards the center of your body from the fovea, or the focus of the eye, and about 1.5 degrees upwards from it. In object space, the blind spot is located approximately 15 degrees laterally and 1.5 degrees downwards from the point of fixation.
The position of the blind spot can be found by placing two dots on a piece of paper and then holding that piece of paper away from your face, covering one eye and then focusing the other eye on one of the dots. At the position where the other dot disappears, mark the distance from yourself to the paper and then use trigonometry to calculate the angle.
So for testing visual acuity, 20/20 vision is normal, which means that a person standing at a standard distance, which is 20 feet, will be able to see a letter on a Snellen chart that subtends 5 arc minutes, but with strokes of 1 arc minute. So you'll be able to resolve the details between black and white of the letter. So if you're reading a Snellen chart from a distance besides 20 feet, you can use this equation to scale your visual acuity. With glasses, Ryan was able to read the 2013 line. With contacts, I was able to read the 2015 line, and Cade was able to read the 2020 line. Without contacts or glasses, Ryan and I can't read any of this. So we've already talked about visual acuity in terms of the Snellen chart with high contrast, but we also have to talk about the eye's ability to resolve objects with varying contrast. The eye's contrast sensitivity depends on the spatial frequency of the object you're looking at. So on this chart, you can see that on the lower end of the spatial frequency spectrum, it's harder to resolve lower contrast values. So the same is true on the other end of the spectrum with high spatial frequencies. So there's actually a spatial frequency in the middle where you have maximum contrast sensitivity, as you can see from this plot.
The eye also has temporal resolution, which corresponds to the minimum time to recognize the difference between two flashed light sources. This depends on the integration time of the eye, where shorter values correspond to better resolutions. And this is also dependent on the different types of photoreceptors, where the rods have a longer integration time at 100 milliseconds, and cones have between 10 and 15 milliseconds. There is also a frequency at which the eye can no longer detect any flickering, which is known as the critical flicker frequency. This is dependent on a number of factors, notably intensity, size, the spectral properties of the image, and where on the retina the image hits. In this lab, we're focusing specifically on the spectral properties. In this setup, we have a function generator hooked up to a circuit board with 3 different LEDs, a red, a green, and a blue. In order to find the critical flicker frequency, we increased the frequency up until we could no longer see the LEDs pulsing. As you can see on this graph, green had the highest flicker frequency for each of us, as it most closely corresponds to the peak of the photopic response as shown on this plot.
The next part of this lab is testing for color vision using a monochromator, or seeing what range of wavelengths that we can distinguish. To do this, we start if we're looking for the lowest wavelength below the threshold and went up, and if you're looking for the highest wavelength, we started above that wavelength and went down. This is because it's better to start from no color and to go to where we can see color, so our brain gets a positive signal as opposed to starting from the color and going away from that. A limitation for the monochromator in establishing color vision is if someone can see beyond 800 nanometers. How the grading works is that the first order of the light and the second order will start to overlap past 800 nanometers, because the second order of the 400 nanometer light will start to be there. So if someone was able to more distinguish longer wavelengths, then they would need a different test or a different grading. Emily could distinguish the longest wavelength at 794 nanometers, and I could distinguish the shortest wavelength at 413 nanometers.